All right, guys, let's do a quick little recap of our biotechnology so far. We know that in order to do a lot of the gene manipulation that we need to do, we're going to need restriction enzymes that are going to be used to cut the DNA at certain restriction points. We're also going to learn today how we can paste the DNA back together so we can insert specific genes into other organisms' DNA. We'll learn how we can copy those genes using plasmids and the method called PCR. And we talked last time a little bit about how we can find certain genes and identify them using gel electrophoresis, which we'll discuss a little bit more today as well. So let's talk a little bit about those ligase and hydrogen bonds in terms of pasting our DNA to create new strands of DNA. So the sticky ends that are created by our restriction enzymes are going to allow our complementary bases to line up. We can see that if I have one piece of DNA cut with the restriction enzyme and I have a different piece of DNA from a different organism cut with the same restriction enzyme, the sticky ends are complementary. And so the T's and the A's are going to be attracted to each other, the A's and the T's will be attracted to each other, and they'll form hydrogen bonds. Once that happens, we create this recombinant DNA molecule. So it's a DNA molecule that has a new combination of genes because we've inserted a specific gene into it. Now the enzyme ligase, which you're familiar with from sealing together the Okazaki fragments in normal DNA replication, is going to do the same job here. It's going to bind our gray strand of DNA with our green strand of DNA to form that entire recombinant DNA molecule. So these are the two important things that we need in order to paste two different organisms' DNA together, or inserting a specific gene into another piece of DNA. So here's a really good example of how biotechnology uses these restriction enzymes. So we can see here that our restriction enzymes are going to cut the DNA at the restriction sites. Here we've got a restriction site and a restriction site here. Now if I use that restriction enzyme, it's going to create these sticky ends, and those sticky ends are going to be complementary. Now, if I use that exact same restriction enzyme from here to cut a different piece of DNA, it's going to create those same complementary sticky ends. And the DNA ligase and the hydrogen bonds can join those two together, and we can create a brand new DNA molecule. So once we've created that brand new DNA molecule, we want to be able to make copies of that new gene. Well, we can do a couple of different things. The first is our plasmid method. Plasmids are small circular pieces of DNA that are found in bacteria. And so we're actually tricking bacteria into copying our DNA for us. The cool thing about these plasmids is that they're separate from the bacterial chromosome. And so they replicate on their own independently of the bacteria's DNA. And so we can use this knowledge to actually get bacteria to copy our new genes for us. So in order to do that, in order to get our bacteria to copy our desired DNA fragments or our desired genes, we need to actually insert them into the DNA sequence, into the bacteria's DNA or plasmid. So here's our plasmid. Now, how do I actually insert it? Well, I'm going to use that restriction enzyme. So here the restriction enzyme has cut the plasmid, so there's this empty space. If I use that same restriction enzyme to cut a different kind of DNA, and then the ligase can be used to join the two together, well, now I have this recombinant plasmid with a new inserted gene. Maybe this gene is used to make insulin, or maybe this gene allows the bacteria to light up or fluoresce under UV light. And that works because the DNA can then be transcribed to RNA, which can be translated into proteins, which result in a trait like insulin, for example. Now, well, how do we get the DNA actually into our bacteria? Well, this is when we use the process of transformation. Transformation is the uptake of DNA by those particular bacteria. So here's our bacteria, here is the cell, here's its normal DNA, and we have all of these plasmids that we've created by and inserted new genes into. And so the transformation process will occur when the bacteria have taken in these new plasmids. The result of this is production of many copies of that inserted gene. And we can use this for a lot of different things. So once we've inserted this new gene of interest into our plasmid, we can then have our bacteria undergo transformation, and we now have this plasmid with the new gene inside of our DNA. We can make a bunch of copies of the gene. That's the first thing that it can occur. And the important thing with making a bunch of copies of the gene itself is then we can use that gene for basic research, understanding the sequences of those genes, or we can insert those genes into different things like plants or other bacteria. And that allows our plants to maybe become resistant to certain pests, or our bacteria to alter their genetic makeup so that they could help us clean up oil spills. So we can give the copies of genes to other organisms and allow them to have new traits. We can also use the plasmid to actually make copies of a desired protein. So a lot of times what we'll do is we'll insert the gene for making insulin into a bacteria, and then we use the bacteria to create a bunch of insulin for us. We can also use that gene to make things like human growth hormone or proteins that dissolve blood clots for people with a heart disease, or we could just do basic research on that specific protein. And so either making the copies of the protein or copies of the gene are two really important ways that we can use this plasmid process of copying. Now, sometimes our fragments of our genes are going to just be too small, and they're going to be difficult to isolate and insert. Or they just won't be able to be incorporated into a plasmid because there isn't a matching complementary base. So this guy here, okay, Mr. Kerry Mullins, he discovered a new process called PCR. PCR is a polymerase chain reaction. It's a method for making copies of a specific segment of DNA. And the cool thing about it is that we can start with just one piece of DNA, and we can make hundreds of thousands of those same pieces of DNA. And the reason we do this, the why, is because we can copy the DNA without the use of bacteria or plasmids. What we're really doing is copying DNA in a test tube. So we need a couple of ingredients. The first thing you obviously need is a piece of DNA to copy. You really only need one cell worth of DNA. So for example, you could collect DNA from a crime scene, or you could take DNA from the inside of a person's mouth, their cheek cells, and get that copy of DNA, and we could put that into the test tube. We're also going to need some DNA polymerase. Remember, the DNA polymerase's job is to add the matching nucleotides onto the replicating strand of DNA. In order to make that additional strand of DNA, we're going to need free nucleotides. Those are the A's, T's, G's, and C's, so we need to have a lot of nucleotides floating around in our test tube so that we have materials to build the new strands of DNA. And finally, we need a primer. The primer is going to tell the reaction, the DNA polymerase enzyme, where to start copying the DNA. And so what's going to happen is we're going to use this thermocycler, and that's going to heat up and cool down the bacteria, or excuse me, the test tube, so that we can get this DNA to be copied. So this is what the PCR process looks like. We're going to mix our ingredients in our test tube first, and we do this series of heating and cooling, and that's going to make a bunch of copies of the DNA. So we can see here that we've got our sample of DNA from a drop of blood. So here's our individual DNA sequence. If we raise the temperature to about 90 degrees, the strands separate. It's breaking those hydrogen bonds. So I don't need helicase to break it apart. I can just heat it up. Then if we cool it down, we start to see that we can have our nucleotides being added as complementary. We heat it up a little bit more to 70 degrees, and the rest of those strands are going to be formed. So we started with one strand, now we have two. We're going to do that again. We're going to raise the temperature. These two strands break apart, lower it back down, they start to form, heat it up to 70, now two strands become four. We do this process 20 to 30 times, 3 steps per cycle. Each step takes about 30 seconds. So what we end up with is millions of copies of DNA each hour, just from one original copy. But we have a problem. When we heat the DNA to denature it, it's also going to destroy that DNA polymerase. So what we could do is we could add a new enzyme every single cycle, but that would be really impractical. Think about it. We're doing this 20 to 30 times. We're not going to add new enzyme 20 to 30 times. That's really impractical. So what we need is an enzyme that can withstand that 90-degree temperature. So what we've done is we've gone to hot springs and we found bacteria that live here in these really hot waters, and they have an enzyme called Taq polymerase, and that enzyme is going to be able to withstand this 90-degree temperature. So what can we do with all these copies? Well, we can run them through the gel to find and compare those DNA sequences and identify suspects in a crime or specific genes that a person may have. We could take those genes that we've made hundreds and hundreds of copies of and insert them into other organisms like plants to create genetically modified foods, or we could use those to just study the gene sequences, the A's, T's, G's, and C's, to understand disease or traits a little bit better. Now, when we run it through a gel electrophoresis, we can actually compare those unknown DNA bands to a standard. And when we compare that, we can determine if a mutation is present in a DNA sample. So we can see that all of these samples have the same, this gene, they all have this gene, they all have this gene, and then there's some differences. Okay, and we can plot these on here to create our standard curve, and we can use the standard curve to figure out what kinds of mutations there are. Are the genes longer or shorter than they usually are? And we can also use this idea to analyze our restriction fragment length, and that deals with polymorphisms. Now, a polymorphism is a difference in our DNA sequence between individuals. So remember, guys, because each person has different DNA, the restriction enzymes are going to cut the DNA differently in each person because everyone's A's, T's, G's, and C's have a different sequence. So the fragments are going to create a different band pattern for each person. Here's one band pattern. Here's a different band pattern for the same gene. So depending on if you have brown hair or blonde hair, for example, your pattern could be a little bit different. And we can actually use these patterns to help us to identify genes or identify people. So here's the first case that was successfully used to try DNA evidence. It was in 1987. So here's our standard that's just showing us that the gel is working correctly. Here's the sample that was taken from the crime scene, and here is the suspect's blood sample. And we can see that the markers line up here and here, and the markers line up here and here. So they have the same genes, it's the same person. Here's evidence from a murder trial. We've got 3 different samples of blood from the crime scene. Here's our standard sample, and here's our suspect's sample. And we can see that blood sample 3 from the crime scene matches perfectly to the blood sample taken from the suspect. Who was that suspect? O.J. Simpson. Found his blood right there in the foyer. So, quick recap. To cut our DNA, we're going to use restriction enzymes. If we want to insert those specific genes into a new organism, we're going to have to paste the DNA using ligase and hydrogen bonds in order to put genes into other DNA to create that recombinant DNA. In order to make copies of DNA, we can use plasmids from bacteria transformation or the PCR polymerase chain reaction to make a bunch of copies of our DNA. These plasmids can then make our specific traits, or we can use PCR and we can then run it through our gel electrophoresis to help us identify genes or people. Hope this was helpful. Make sure that you wrote all the notes down. See you guys soon.